Alrighty. Hello, my name is Daniel Trujillo, and I am presenting my potential projects I will be doing while receiving a master's here at Walla Walla University. Now, in order to understand the project I plan on accomplishing, we need to first define its context, and that context being growth to development. So what is growth to development? Well, it is an essential function that allows cells to transition from single cellular to multicellular. And generally speaking, these are both multi-factor stimulus as well as multi-gene stimulus, which encompass a very diverse range of genes. Additionally to this complexity of stimuli, in general, this transition is induced by an outside factor. This outside factor can include lacking of nutrients, changing in optimal pH levels, or even changes in optimal temperatures. So if we can think big picture for a second, let's look at one of the most classic and arguably the most important examples of growth to development pathways, human development. And as you might know, humans start to develop the moment the sperm enters the ovum. From there, cell division takes place. And following that cellular division, the cells receive signals to communicate differentiation, resulting in complex structures like organs and tissues. Now, the main workhorse of that development is stem cells. These stem cells are essentially blank canvases that tell the entire system to become specific cells, like organs of like heart tissue, heart cells, heart tissues, organ or lung cells, stuff like that. Now, with that, with those stem cells generally stay within us and assist in little homeostasis processes like organ/tissue repair. But as these are all positive processes, there are a few negative processes that happen through the similar cycles of development, and those would be cancers. So in these situations, cancer is a result of cells that are told to develop and they go through all the right checkpoints, but then they do not die when they're supposed to die, and they result in a tumor and causing cancers that spread. Now, in Dr. Lindsay's lab, we have both talked about projects that we could do. He really likes development, and he really likes human development. He really likes cancers research. And well, if I'm working under him in a perfect world, if I want to know more about human development and human cancers, I should be using human cells and human embryo cells. But that probably isn't very cost effective, nor is it very legal. So, and we need to have a model system. And so in order to find a model system that would be relevant to human development transitions, the model system would more or less need to have some specific criteria. These criteria would include a relatively low number of encoding genes, because I personally do not want to dive through thousands and thousands of genes just to find one problem. In addition to that, these genes would need to be existing in a haploid chromosomal arrangement. For humans are existing in a haploid chromosomal arrangement. So if I want to apply this, what I find to humans, we would need to be kind of on the same page there. In addition to that haploid chromosomal arrangement, the pathways of development would need to be taking place very similarly, or at least using the same genes, receiving the same signals, resulting in a similar cellular response. So what model system do I use, you ask? Well, Dictyostelium ductium would work very well. So who is Dicty? Well, Dicty itself is a soil-dwelling amoebozoa that's consuming bacteria and yeast naturally in the wild. Dicty's whole genome is only broken up into 13,000 encoding genes among 6 chromosomes, so it's relatively small, not as big as humans, but not as short as maybe a roundworm. And this whole the way it develops is that it transitions from a unicellular mass to a multicellular organism. And that process is initiated by starvation, something we'll dive into a little bit later. Now, with that whole developmental cycle, it's relatively short, for it only takes place in about 24 hours. So great. we have this slime mold organism, but why should we specifically use it for my projects? Well, first of all, amoebozoas represent a common ancestry of all eukaryotes, and is probably one of the last common ancestors we still have. And with that common ancestry, phylogeny shows that amoebozoas derived from the animal fungi lineages after an animal plant split. Yet Dictyostelium specifically retained a lot of the diversity that's still seen in the eukaryotes. And in addition to that ancestry, the haploid nature of the dicty shows that any mutagens that happen are very easily observable. And I guess as far as lab trials, this very short developmental cycle creates very easily repeatable experiments. All right, so here we're going to take a closer look at exactly how this transition takes place. And it's, as you can see on the right here, we have a clock. This is taking place over a 24-hour time frame. And each little step here is a portion of this developmental cycle. So here at the beginning, we have this little guy. He's happily eating on bacteria and yeast, and he's dividing via binary fusion. Well, once he eats everything that's there, starvation happens, as you might assume. And as those starving cells, as they cells start to starve, they start to secrete CAMP. And CAMP stands for cyclic adenosine 3,5 monophosphate. And this molecule is signaling these cells to start to aggregate and experience differentiation. So as we're moving along here, we're receiving CAMP waves, the little dictyostelium is expanding, and then it eventually forms this mound where two types of cells form. These are both pre-spore cells and pre-stalk cells, where this very little tiny tip here is further CAMP secretions. Now, as time progresses, these cells start to differentiate, causing the whole structure to elongate, where these cells will then start to become their specific places. Now, here at this little halfway point, we create this slug where our cells are now becoming more stalk-shaped and more spore-shaped here. And then as we move along here, we're getting further along in the cycle. The cells differentiate to become stalks and then fruiting bodies. And then the whole cycle ends with a fruiting body up here, then leaving the whole structure and the rest of these stalk cells dying. So this is pretty standard cycle for how this guy functions, how he lives. But these specific places of interest are these CAMP waves. And so let's take a closer look at exactly what's happening during these CAMP secretions. All right, so this is a very complex process here. Not everything is relevant to the projects that I'm trying to accomplish, but they'll give you some more insight here. So I'll point out which ones are most important. So here we see our CAMP cycle. It is a positive feedback loop. For CAMP is being secreted inside the cell as well as outside the cell, and thus causing this whole cycle to take place. Now, the first thing that causes CAMP to start secreting inside the cell is this gene ACA. And so that is adenyl cyclase. And this causes CAMP secretions to start right at the initiation of starvation. Then the CAMP is excreted outside the cell, where it then binds to the CAR-A or CAR-1 receptor. Then that allows all of the rest of this cycle to continue after that binding of CAR. So in addition, something that's not seen here is that these CAMP secretions allow for the activation of the CASA-A gene, which allows for the cell adhesion to take place. So as I mentioned on this previous slide, this aggregation separates the cells and causes them to all come together with that adhesion. And that adhesion then forms these mound structures for later cell differentiation. So In addition to this pathway here, there is another motive force that is assisting in this protein expression. And that motive force is ubiquitination. So what ubiquitination is, it is a pathway that is involved in the changing of protein expression by selectively degrading specific proteins. And specifically in DICT, it is broken up into two phases. So here we're going to see, indicated by number 1, is the phase 1. Here we have ubiquitin existing freely. Here we have a substrate protein. And these two are going to come together by these various enzymes in the ubiquitin pathway. So first, these come together with the E1 activates the ubiquitin. The E2 enzyme then transfers the activated ubiquitin to this E3 enzyme, which then forms this ubiquitin intermediate. They all come together. And then this intermediate substrate is then tagged with this polyubiquitin chain to then be transferred to the phase 2. Now this phase 2 is using a specialized 26S proteasome, and that's what you see here underneath my number 2. Now this 26S proteasome is composed of two 19S regulatory proteasome complexes and a 20S core particle. Okay, so here's our 2 up here. And here is our 20S core particle. So that 20S is formed by 4 rings, 1, 2, 3, 4, stacked together into 2 alpha rings and 2 beta rings. So as this tagged protein is received by this proteasome, it moves through it. So that 19S recognizes the tagged protein and unfolds it by passing it through those alpha rings present in the 20S core particle. So it moves on through here, these two recognize it, then they move it through here. Then as it moves through there, these polyubiquitin chains is then released, and then it is deubiquitinated. It is disassembled. Now, this deubiquitination is a very important enzyme, and it is specifically known as a DUB. So as their name suggests, DUBs catalyze the hydrolysis of an ester, diester, and amide bonds at the C terminal of the glycine 76 of the ubiquitin. So it takes off ubiquitin off of a protein. And so these DUBs may either disassemble the free polyubiquitin chain or remove it completely. Yet most DUBs, however, just detag proteins for degradation that should not be degraded under the present cell conditions. Okay. So now these DUBs are important. Specifically to Dr. Lindsay is a UBPA DUB that is essentially very similar to that of a homologous IOST in humans and the UBP14 in yeast. And what this DUB is directly disassembling those free polyubiquitin chains, as we saw, and it is present during both growth and development, and as we'll go into later, is exactly required for development to take place optimally. All right, so since we have something that we know is needed, what would happen if we take it away? Well, Dr. Lindsay created a mutant that does just that. So these are cells that completely lack the DUB UBPA. And the result of bacterially grown conditions, so normal, what they do in the wild, these UBPA minus cells grow, but they do not aggregate. So as I mentioned in that pathway, aggregation is what then leads to the cell to then differentiate. But if they do not aggregate, they don't form the stalk or the fruiting body, and it fails and dies. So then he looked at cells that are grown under concentrically grown conditions, and these cells do aggregate, but they fail to form these mature fruiting bodies. So they form these kind of flip-floppy shapes that aren't necessarily any representation of what the cells should look like. So we have a problem here. I guess additionally, the UBPA cells aren't producing CAMP or aren't receiving CAMP, as we know that CAMP is necessary for the aggregation. So you can add that CAMP in later and they will grow, but again, they still form these flip-floppy little structures. All right, so here's our problem. Our cells are broken. And I necessarily do not think that UBPA is the only cause of this problem, for there are some key gene players that are working in tandem with UBPA to assist in development. So these genes include LMCA and /B, which is sensing nutrient levels throughout the entire cycle, as well as YAC-A and PUF-A genes, which regulate the transition from growth to development. Specifically, YAC-A deactivates the PUF-A gene. Now, as I mentioned earlier, that ACA gene that secretes CAMP is one of the earliest genes expressed following starvation, and that ACA produces intracellular and extracellular CAMP that drives further differentiation along and morphogenesis of the dictyostelium cells. All right, so now we get to my project, and my project is broken up into 2 and potentially 3 parts. This first part would be gene-dependent saving experiments, so taking some of those genes I mentioned earlier and implementing them back into our mutant cells. Our second project would be to taking a normally working UBPA cell and doing a protein pull-down experiment to see all of its working parts. And then my third potential project would then be to take those Dicty cell mutants and see if we can save them based on the type of nutrients that they're living in. All right, so let's do a quick overview of the rescuing of the UBPA mutants. Well, the goal is to save the mutant by reintroduction of the genes that have been potentially deactivated with the removal of UBPA. So it'll look something like this. So as a control, we would take our UBPA minus cells and then re-implement UBPA. That has been documented to save them. We would see proper growth. Then we would take some more mutants and implement both LMCA, LMCB, YAC-A, and potentially ACA. I guess an important thing to note right here is that ACA, we don't have direct methods of how to extract plasmids of that, so that is just a potential part of this experiment. So how do we get these LMCA plasmids? Well, to begin with, the sequence needs to be cloned into PDX and amplified with E. coli host cells. From there, the PCR products would need to be digested with the enzymes HIN3 and PST1, and then purified with the QIA quack gel extraction kit. Then our reaction volume will be roughly about 30 microliters, and that would consist of DNA, plasmids, buffers, and ligase. From there, our mixtures will then be transformed into a DH5 alpha E. coli cells via mini preps. Now, for our YAC-A expression plasmids, we're going to have to do a kind of different step, not like the LMCA. So what we need to do is create a PUF-A knockout mutant, which would then lead to an overexpression of the YAC-A plasmid. So to do this, we use a P84 eco that would be used to disrupt that PUF-A by homologous recombination. From there, this plasma would be linearized with echo R1 to allow for gene replacement by homologous recombination. Then the linear knockout plasmid would replace the section of the chromosome containing the PUF-A gene, resulting in YAC-A plasmids. All right, so we got both our plasmids. Now it's time to do something with them. So adding them into those mutant cells. So we would take those cultured mutants and then plate them by centrifugation. From there, the cells would be washed twice with cold H50 buffer, which would include all of these little things in here. I'm not going to go into it for this kind of redundant, and then they would be resuspended in more of that H50 buffer. From there, the plasmid DNA would then be mixed with those Dicty cell mutants and electrophorated in pulses. From there, the cells plus the plasmid mixture will then be incubated on HL5 medium and culture the plates for 24 hours. So all in all, this whole experiment will have a few various data analysis to see if things were successful. The first would be just visual observation. Did they grow? Did they form their fruiting body and their stalk? Did they not? Did they form those flippy floppy kind of structures? We would also need to use Western analysis, since we are using PCR products, to see the protein expression. And then we'd also use Northern analysis to see some mRNA expression, to see all the little guys if they're either activated or expressed or deactivated, and there's lower expression. We need to see what's going on there. We would also need to use an SDA polyacrylate gel electrophoresis analysis to see if we added the genes, were they even transcribed, are they present when we see it as far as looking at everything present in a gel, can we see little lines and bands that it was read, it digested it, everything worked. And then our expected results would be a linkage of the gene and UBPA interactions, revealing specificity in the growth to development pathway, thus fixing the dysfunction. All right, so the second part of my project would be the UBPA protein pull down. So the goal of this is to identify the proteins that are directly interacting with UBPA. Now, the methods in order to achieve this, first we have to identify the interacting partners of UBPA, and to do that we have to use a tap tag UBPA expression plasmid. And this tap tag is specific for the UBPA. So the UBPA coding sequence will be then cloned into this PC-tap or a PN-tap expression vector. From there, the UBPA coding sequence would then be amplified by PCR, and then those reactions will contain the cDNA, the PUF Ultra 2 fusion HS DNA polymerase, a PUF Ultra 2 reaction buffer, a DNTP, primer, which we haven't specified yet, and water. From there, the primers will be digested with restriction enzymes, ligated, incubated, and incubated with E. coli cells. Based on the restriction enzymes used, maxi preps will then be prepared and analyzed for DNA sequences via gel electrophoresis. Then the samples will then be submitted and subsequently compared to the sequences in the DICTI genome. So I guess I mentioned a little earlier, the gel electrophoresis will verify that we have a tag on there and that we see everything in there. Then comparing to the DICTI sequences on the online would show what our UBPA is interacting with. We can do side-by-side comparisons and see what's being transcribed. Then our expected results would hopefully reveal all those interacting proteins involved with nutrient sensing, transitions, stuff like that. So that about concludes what I plan to do as far as a project here. And as I mentioned earlier, this gives us relevance to this further understanding of the growth to development cycle. It'll give us more relevance to the direct gene interactions required for that cycle. And then it would also give relevance to the proteins required for that transition. And hopefully we would fix Dr. Lindsay's mutant to then allow for further projects later on to see what further things can happen if you remove specific things in this process. Thank you very much.